Я справді вірю, що конференція – це лише початок.
Сьогодні науковці працюють в дослідницькому середовищі, яке їм надає можливості, про які ще 10 чи 20 років тому можна було тільки мріяти. Ми вперше можемо на багатьох рівнях, починаючи від епігенетичного і закінчуючи системною поведінкою організмів, розуміти і інтерпретувати живі системи, відкривати нові явища і розробляти нові терапевтичні засоби.
Наука – це марафон із величезною кількістю перешкод. І потрібно бути наполегливими, готовими до великої кількості проб і помилок і сміливими для того, щоб втілювати нестандартні підходи. І тоді результат вас здивує.
Hello everyone and welcome to BioGENext series. We are really thrilled to see so many people here right now.
It is a real pleasure to see so many people interested in the practical aspects and applying advanced methodology in life science research, joining us both online and in person today.
But before we begin, let me introduce my wonderful co-host, Dr. Oksana Mankowska, dedicated researcher at the Department of Molecular Oncogenetics at Institute of Molecular Biology and Genetics of National Academy of Science of Ukraine.
It is my...
Yeah.
Thank you. It is my pleasure to introduce Alexander Huber, head of the Biotech R&D at YP Biotech, Biopharmaceutical Research Unit of Urea Farm. We are excited to share with you not just what we will see today, but also tell you a little bit more about what this Green Series addition to the BioGENext logo really means and anticipates.
But first of all, we truly want to start by expressing our deepest appreciation to the armed forces of Ukraine, to all of the defenders of Ukraine, to every person who bravely stands for our country, for each one of us here, and for humanity as a whole during these 3 years of brutal Russian aggression against Ukraine. Only thanks to them we are able to be all here and working towards the development of the future of Ukraine. So now maybe ask all of you to stand up and observe a moment of silence to honor the memory of all the defenders who devoted the most precious thing each of them had, their lives, defending not just our country, but all of the values upon which free words truly stands. Thank you all.
BioGENext is an initiative created by scientists for scientists. About 1.5 years ago, our teams from Europe Farm R&D and the Institute of Molecular Biology and Genetics, National Academy of Sciences of Ukraine, decided it was time to combine our efforts towards a very important goal, to build a strong, thriving biomedical research community in Ukraine and create a platform that will enable academia and industry to meet each other, learn from each other, and unite all the best expertise and resources available to effectively collaborate on enabling research for next-generation therapeutics.
And the vision of such a platform has materialized into the BioGENext initiative. Last September, the initiative started by hosting the very first BioGENext conference here in Kyiv. It was really inspiring event, gathering more than 400 people in person, with additional 250 persons joining us virtual. We were honored to host more than 40 speakers sharing their expertise in various research areas covering both therapeutic and diagnostics development, and inspiring us pursuing cutting-edge technologies and research here in Ukraine.
This conference became a starting point, catalyst, if you will, providing not just knowledge, networking, partnership, and collaborative projects, but also inspiration and new perspectives on our own research for many of us here today. But this was only the beginning. In numerous interactions with both academic and industrial scientists, we saw a great demand not only for a platform to collaborate around the key research initiatives, but also for an environment that allows discussing various methodologies, practical aspects, and the implementation, and gaining feedback from experts in the field with extensive hands-on experience.
And today we're actually very excited and happy to launch the project aimed at exactly that. BioGENext series actually is a spin-off educational initiative jointly created by Institute of Molecular Biology and Genetics, Farm, NGO Unia Scientifica, and YP Biotech. And this initiative is actually specifically designed to empower you, both current, active, and future researchers that perform development work in Ukraine. The main aim of the initiative is actually to help you master and increase your proficiency in some of the cutting-edge methodologies or even state-of-the-art methodologies from experts and that are actually working on the development of such kind of methodology and their implementation on a daily basis. This new experience and knowledge will actually give you a new perspective on how to tackle your own research problems, improve the quality, probably will improve the quality of implementation of some methodology that you're actively working on in the lab by deeper understanding what you are doing, what are pros and cons of choosing a specific methodology for your tasks. And as a result, it will definitely boost your impact of your research later on.
The BioGENext series will be structured as monthly public lectures, each lasting around 2 academic hours, with an extended Q&A session, where all of participants can, and I encourage to, of course, ask the questions related, not only directly related to the topic of the lecture, but also the questions which addressing the relevant aspects of their own research or ongoing projects. The lectures will have a hybrid format, and we stimulate participation in Q&A session of the participants from online, asking their questions and leave chat during the stream. You're welcome.
And what is actually really unique about BioGENext series is our aim to bring advanced and practical knowledge directly to the universities and the research institutions. Thanks to the numerous academic partners and their active contribution to the initiative, next 3 lectures will be held probably in your very own alma mater's large lecture hall, helping you to learn some critical skills and gain some specific and very important knowledge about methodological applications directly in your academic environment. Moreover, this format will give you the opportunity to create connections with some of the local research groups that are working in different institutions, which we hope will lead to new collaborative projects and a bold impact.
The lectures will focus on the state-of-the-art or emerging methodologies ranging from CRISPR screens to multi-omics and organoids applications, everything you can see here, and all of which typically require input from experts for correct and effective implementations. These lectures aren't just theoretical. They'll be focused on developing of a set of knowledge and skills required for a deeper understanding and effective implementation of these advanced methodologies in your research projects. And if you feel the society is particularly interested in some additional topics, we will be happy to hear from you.
So we truly hope that you will join us not only on this lecture, but on the whole wonderful journey which is BioGENext series actually is. Because it's quite a unique chance to, first of all, gain a new valuable knowledge and expertise in some of the methodological aspects, broaden your professional networks, and as a result, later on, actually to contribute to the development of the creation of some transformative medicines or diagnostic solutions. So we are really happy to see all of you here, and let's get started.
Today, we are excited that our inaugural BioGENext series lecture is held at such inspiring location as MacPau Space. And it's hard to imagine another public space in Kyiv that aligns better with the ideas with BioGENext series. Many thanks to MacPau Space, to all the partners, to all organizing team, and to everybody who contributed to making the BioGENext series a reality.
Yeah, and before we begin, let us inform you that your safety is actually a primary priority for our team. So in case an error alert will be announced, we will pause the event and we will ask you to move into the shelter located downstairs in the same building. So in this case, please listen to the communications and announcements from the organizing team and follow the MacPau Space personnel that will guide you directly to the shelter. The shelter is fully equipped with all the necessary presentation equipment, and as soon as all of us relocate there, we will be able to continue the lecture and Q&A session.
And now, let's dive straight into the first lecture. It's my great pleasure to introduce today's moderator, Dr. Natalia Shenderovska, a senior researcher at YP Biotech Therapeutics Research Unit at Urea Farm. Having earned her PhD in virology, studying the structure and molecular biology of bacteriophages, for the last 4 years at YP Biotech, she has been leading research programs focused on the development and implementation of CRISPR systems for engineering eukaryotic protein producer cell lines. Let's warmly welcome our moderator.
Thank you, Oksana. Thank you, Alexander, for this kind introduction. I truly hope that BioGENext series will become a uniting place for the Ukrainian biomedical community. And today it's my great pleasure to introduce our first lecturer. It's Dr. Andrew Bassett. Dr. Bassett leads the cellular and gene editing research at the Wellcome Sanger Institute. He focuses on the development of genome engineering technologies and cellular differentiation methods with a specific focus on neurodegenerative disorders such as Alzheimer's and Parkinson's disease. His work includes the developing methods for scaling genome engineering protocols, modulating the epigenetic and transcriptomic states of the cell, and improving the efficiency and specificity of genome engineering technologies, which you will actually hear about today. Prior to joining the Sanger Institute, Dr. Bassett earned his PhD degree at the MRC Laboratory of Molecular Biology, researching chromatin remodeling in heterochromatin formation. He furthered his postdoctoral studies and actually was one of the pioneers in developing the CRISPR technology to be used in Drosophila. And in our personal communication, he kindly offered that the participants participants of BioGENext series are welcome to ask any questions which can go beyond the scope of his lecture, so that he could provide more insights and give practical advice, which you will be able to use in your own research, in your own labs. So now, please be active, take this chance, and let us welcome Dr. Andrew Bassett.
Hello. I hope you can hear me. It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening. I'm going to start off with something very basic. I think all of you will probably have come across the CRISPR system. And this was something that was now around about 10 years ago or so. It was actually a little bit more than that, 2013, that it was kind of applied to eukaryotic organisms. And it's actually as with many scientific developments, it's sprung out of something completely unrelated in terms of why yogurt bacteria actually became resistant to bacteriophage infection. But I think the realization back in 2012 of the fact that actually this was a RNA-guided mechanism for targeting viral DNA was actually the real breakthrough from this point of view. And actually, it turns out that what happens is the bacteria remembers whatever viruses have infected it before and puts those sequences into the genome as little short segments of about 20 nucleotides separated by a common repeat in between. And those act as sequence-specific guides to target a Cas9 nuclease to the viral genome. So I think back in 2013, it was realized that actually you can understand the specificity of this much better than you could with any of the prior technologies because it's simple base pairing rules. So essentially you can then repurpose this nuclease to cut anywhere you like in the genome just by simply changing this first 20 nucleotides of sequence, which was remarkable really, because then basically you can essentially target anywhere that you like in any genome, actually, it turns out, including human genomes, just simply by providing this protein and this RNA with changing this first 20 nucleotide sequence. Now, that's obviously very useful for making cellular models of disease, knocking out genes, et cetera, but I think very soon afterwards, it was realized that actually these 20 nucleotide sequences can be synthesized on oligosynthesizers, essentially. So in a similar way to what was done with microarrays, you can actually assemble many thousands or tens of thousands or hundreds of thousands of these different sequences to allow you to target every single gene in the genome. And that's basically what I'm going to be talking about today. So these were some of the very early papers that came out on this, where actually people designed, in this case, around about five of these guide RNAs per gene across the entire genome. And then they were able to use these to perform forward genetic screens in human cells. And this was really a breakthrough in that kind of field, because forward genetics prior to that point had been limited to flies, really, or in some instances certain weird and wonderful cell lines, yeast and various other things. But this actually allowed us to be able to do these forward genetic screens in cells on a genome-wide scale. Now, I wouldn't necessarily recommend you use any of these libraries now. They've all been superseded, and I'll go through a bit more about that later on. But I think it's just to highlight that this technology has now been around for quite a long time, and it's been widely applied across lots of different applications. So the basic principle behind a pooled genetic screen using CRISPR is that you clone this population of maybe 100,000 different guide sequences. It's normally a viral vector, so this is normally a lentiviral vector, and such that each virus has a different guide sequence within it, and then you infect those into a population of cells, and you do this in such a way that you lower the amount of virus such that each cell takes up one and only one of these vectors. Now, because lentivirus integrates into the genome, that also integrates these guide sequences into the genome, and they act as a marker of that particular cell. So you then have to select these cells because obviously in order to get this to be one infection event per cell, you actually have to have quite a lot of cells which don't have an infection event. So you select for those that do actually have the infection event, normally with some drug selection or fluorescent selection. And then you end up with a population of cells, each of which is labeled with a different guide sequence that's integrated into the genome as a marker of what it is. And that guide will target a particular genetic knockout within that cell. So what you essentially have is a population of millions of cells, each of which has an independent knockout of any one of 20,000 genes in the genome. Now, that's an amazing resource if you think about it. If you have some kind of way of sort of measuring that, which you can do by high throughput sequencing of the guide sequence, you can then expose these to various different sort of selections or stresses or various other processes, which we'll go into in a bit. And the simplest of those is just to grow them. And you can imagine that sort of, for instance, if this orange cell had knocked out a tumor suppressor gene that enhanced its growth, then this cell would outcompete the other cells within the population, and you'd end up with more orange cells. Similarly, if this purple gene had a growth deficit, then this cell wouldn't divide or would die off, depending on how bad it was, and therefore that would eventually sort of disappear from the population as the other cells around it grew. So then what you do is you over the guide sequence you do a PCR for the guide RNA across time, and then you quantify the proportion of the different guide sequences at time 0 and then at various time points after that initial point. And then you can do sort of comparisons between all these different sort of time points to say, well, this red guide is outcompeting and this purple guide is depleting, and that gives you a measure of the essential genes and the tumor suppressor genes that might be affecting that particular cell population. So this has been heavily applied, including a lot of work at Sanger, not, I should hasten to add, by our group necessarily, but we have been involved a little bit, to generate this cancer dependency map or DEP map. And this has actually involved doing genome-wide screens across now nearly 800 cancer cell lines, which is almost the sort of total of the 2D cancer cell lines in the world, and is now being extended into into more complex sort of cancer models and other cancer models. And I thought I'd just illustrate sort of, how powerful this kind of methodology could be. So this is actually some results from a few years ago now from that study, where each of the dots on this graph represents a gene that, if you knock it out, affects growth of at least one, or in many cases above this threshold line, multiple cancer cell lines within that data set. And these have actually been grouped according to kind of, whether there's an approved drug already existing for them, whether there's drugs in clinical development, and whether there's kind of not so much known about them, essentially. And you can see that there's a lot of the sort of approved drugs for cancer therapies that are already in there. Also some in clinical development, but there's also a huge amount, all of this sort of load of genes up here that are actually potential candidates for therapeutic intervention in terms of various different cancer types, and they've been coloured by their sort of effectiveness on different types of cancer cell line. And there was one of these that this Werner's gene, which was followed up in a bit more detail, and actually is now in clinical development. And this actually turns out to be a helicase, not necessarily expected at all to have an effect on cancer survival. But it turns out that if you look at, this is the control, so these are non-essential genes. These are essential genes. So there's a big loss in viability when you knock out essential genes compared with non-essential genes. And actually, you can see the same effect with knocking out of Werner's in these microsatellite instable sort of cells. So this is a particular class of colorectal cancer. It doesn't seem to affect other classes of colorectal cancer or indeed normal cells. So this is actually an incredibly promising candidate for therapies. And they were able to show in mouse models that actually you can by inhibiting this or knocking this gene out, you can actually prevent tumor growth and division. So actually, this has now been taken on by a couple of different drug companies, and then they're now developing this into a therapeutic, which will be going into trials sort of soon. And we're involved. I think it's worth mentioning that we're involved in some extensions of this, and sort of now, obviously, the kind of single gene knockout space has kind of been looked at in quite a lot of depth. We're now moving into kind of other things like dual perturbation screens, and this is just some work where we've actually put two guides separated by a tRNA spacer. And this gets cleaved out in the nucleus by RNases and generates two independent guides, which then, because this is quite a short construct, we can actually clone these as libraries of pairs of guides. And that means that then we can look for genetic interactions. And this is just an example where these are actually paralogs within the same species, that if you knock each out individually, you don't see any effect on growth, but when you knock them out together, you see a strong decrease. And now this is being used a lot at Sanger to kind of screen for synthetic lethal combinations or pairs of genes that when you knock them out together, you end up with loss in viability. So that's, I think, a nice illustration of why this is important and how useful these kind of screens can be. And I'm going to go through sort of a bit more about the kind of the nuts and bolts of how you go about these screens and things that you'd need to watch out for. So this involves lots of different steps. So there's obviously a lot of different ways that you can perturb the cells in the first place, including the knockouts that I talked about, but also these different sort of other methodologies for downregulating or upregulating genes, looking at dual perturbations, even SNPs and overexpression, which I'm not really going to talk about, but if anybody's interested, please do get in touch. Then there's kind of what cell model systems you might want to use this in, because that has a big impact on how easy and how you would actually go about these screens, and also what readouts you would use for this. And primarily I'll focus on these upper readouts here today, but just to note that we're also heavily developing these kind of more complex readouts, such as single cell transcriptomics or imaging based optical pooled screens. So starting off with the perturbations, again, I'm not sure sort of what background everybody has, but essentially CRISPR knockout is using the endogenous sort of form of Cas9, where it makes a double-strand break, and that often gets repaired so that you end up with small indels, and these will shift the frame of a protein-coding gene and result in a knockout. But you can also remove the catalytic activity of Cas9 with a couple of point mutations. And that then allows you to use that as a sequence-specific DNA binding factor. And then you can fuse onto this various different transcriptional activations domains or transcriptional repression domains and actually manipulate gene expression levels in the cell. So the three that have been most sort of heavily used are knockout, which is what we mainly focus on. And I think this has some advantages in that it actually gives you a full, complete knockout. And also, it doesn't necessarily require a maintenance of Cas9 expression. But there are some sort of oddities to it. So in terms of the fact that it does generate a mixture of different alleles. So these indels are semi-random. So you'll end up with a 1 base, 3 base, 5 base deletion. And some of those may or may not give a knockout of the gene. So there are certain sort of disadvantages with it, but mainly it's been working very well in our hands. And just to highlight that there are alternatives. So CRISPR-I is a way of sort of downregulating genes. If you target their promoters, then you get sort of good knockdown of both the RNA and the protein. But that does require this maintenance of Cas9 expression, and it is sort of promoter specific. And what I mean by that is that you, if you have multiple promoters for a gene, you can only necessarily repress one, and that may not be enough to actually obliterate its function. So, and also it might not be a complete 100% knockout. So that can have its advantages or disadvantages depending on whether you're sort of, you're looking at essential genes or not. But that is definitely a disadvantage with that methodology. And CRISPR-A is the converse. So this is probably one of the few ways that you can screen for upregulation on very large scale, anyway, genome scale. And again, suffers from some of the same sort of restrictions that the others do. I think it's worth just highlighting that, I mean, we've been looking at a lot of these other more complex editing sort of events, so things like the dual perturbation screens, which is working quite nicely, but also SNP screening using things like base editors and prime editors. And essentially the issue with those at the moment is that either the editing is not super precise, so actually you end up with quite a lot of undesired edits, and therefore, this is in the case of base editing, where it's hard to deconvolve what's actually the causal sort of SNP that you're introducing and what's a bystander. And prime editing is very attractive, I think, but it is still not very efficient, and it's the design rules for the guides that you need are not terribly well understood. So it's not really feasible to use that for screening successfully, and unless it's a positive selection screen. So the second thing about sort of, your kind of choice of what to do is also down to sort of how you deliver your components of the system. And there's kind of two options here, really. One is to use an all-in-one vector. So this is actually where you deliver your guide RNA and your Cas9 protein on the same vector. Now, the issue with Cas9 is that it's quite big, and therefore an lenti-vector has a particular carrying capacity, cargo capacity. So actually you're getting very, very close to the cargo capacity by the time you've put the guide and the Cas9 on the same vector. And this means you're, whilst it's very attractive because you can deliver everything in one go, and that means you don't have to produce any Cas9 lines, and you can deliver it whenever you like, because lentivirus tends to infect most cell types. It does restrict you in terms of your promoter choice, because you're limited in cargo capacity, and also the selectable markers that you can use for selection of this. So and it's also right at the limit of the viral packaging, so that means that the titer of the viruses that you get is lower, although that is definitely solvable. So the other method, which people often use, is actually this two-component system where you deliver Cas9 on one virus, or by transgenically introducing it into a safe harbor site, for instance, and you make a cell line that contains that Cas9. And this is how all of those cancer cell line screens that I've described were done. And then subsequently you introduce your guide library, which is just a guide library by itself. That means you can then put multiple selectable markers, in this case a puro and a fluorescent protein. And that means that sort of you can then select for those very, very effectively. It also has the advantage of being a little bit sort of safer, because this means that you're splitting the process into two halves. And there are some sort of modifications of this, for instance, using Cas12, which is a slightly smaller protein, which I'll go into in a second. So the next thing is actually coming back to what I said originally, really, about sort of some of those original libraries not necessarily being the best ones. There's actually a huge number that are available for genome-wide screens. And if you're interested, this is a great place to look, the AdGene website. This is where most of these are now deposited, and it's been an amazing resource for the scientific community, I think, to be able to just order these up for very sort of relatively small amounts of money. And this is actually from a review paper a little while ago, just looking at the sizes of these different libraries. So the first generation, I would say, of libraries, or maybe the second generation of libraries, were things like this Brunello, the TKO, the Kasuke-Yusa, which is the one from Sanger, and the Vienna library. And these were all using single guide delivery and looking at having round about three to five guide RNAs per gene. So that means that for a 20,000 gene genome, you have around about sort of somewhere between 80,000 and 100,000 guides. And this obviously is very powerful, and this is again what I've shown previously is very successful in these screens. But if you look at sort of screening this kind of size of libraries, you need around about 30 million cells per repeat, or about sort of 100 million cells if you're doing sort of 3 repeats of a screen, which is actually quite a lot in terms of various different bits and bobs. And I'll explain sort of how this doesn't seem like a lot when you're thinking about just growing cells in a dish, but there's a lot of losses in the process. So actually this translates to having a lot more than this to begin with in many cases. So there has been a push to kind of reduce the size of libraries, and there's been sort of a couple of different sort of attempts at doing this. So this minlib is one that I'll explain in a minute, and this inzolia is another one. But I think it's worth just highlighting sort of, where some of those losses might happen in the screen. So this is what I've shown previously. And if you imagine that you need to sample enough cells to give you a reasonable readout. So let's say that you had 100 cells at this early point for this orange mutation and 100 cells for this purple mutation. And then you follow them over time. You could then look for changes, both positive and negative, in those numbers of cells. If you went down below that, you start to end up with a lot more stochasticity in it. So one of the big things about any screen that you do is actually to keep the coverage definitely above 100. That's the absolute minimum that we've kind of found to be reasonable for sort of things like these cell death, cell viability screens. And in many cases, this needs to be much higher. You need to be up in the sort of 500 times coverage for the library. And this is to account for sort of the stochasticity of certain cells depleting, but it's also to account for the fact that the guides aren't evenly distributed across the library. So some of them will be represented much more than others, and therefore you have to be able to account for that in that experiment. And then there's particular pinch points, I guess. They're kind of bottlenecks in the process, and I've highlighted a couple here. I'll illustrate it with a particular example later on. But one pinch point is post selection. So you can imagine you start off with a certain number of cells, you're transducing them to get a single copy integration, and in order to do that, you have to only have about 30% of the cells being transduced. And therefore, after puro selection, you've actually reduced the number of cells by roughly 3 times. So that often is one of the pinch points. And the second is actually when you're kind of doing the final step of sort of DNA extraction and PCR, you only have a single copy integration of your lentivirus per cell. And when you extract genomic DNA, that's often quite a lossy process, and you lose quite a bit of DNA or lose some of the complexity there. So when you, and also there's roughly 5 picograms per genome. So per 5 picograms, you have 1 copy of your lentiviral integration. So that means that in order to sample from 1 million cells, you need actually about 5 micrograms of genomic DNA. And that means that often people don't do enough of this kind of this PCR reaction. So you typically can only put in a microgram or so in each PCR. So actually, for a typical screen of maybe 50 million cells, you'll need sort of something in the order of a plate or sort of 2's worth of PCRs to actually be able to do that. So it's very easy to lose at that point, and also lose in the kind of PCR efficiencies that you have in that step. So there's a lot of little technical points here that actually matter a lot in terms of maintaining that coverage. And you can see how actually, if you need this very big number, you're looking for the bottleneck at whatever point that bottleneck comes as being that coverage point that you're looking at. So that's, I guess, just to sort of highlight the numbers, and there is also a sort of a number of different libraries available. So obviously, sort of vector number is one component of that, but there's also different designs in terms of how they've been put together. This is actually quite a nice paper from fairly recently that did a very systematic comparison between the different libraries. And you can see this is just looking at the loss in viability of essential genes across a few different cell lines with different libraries. And you can see that things like the gecko library, this red line, which is one of the early ones, doesn't perform very well. Most of them sort of sit pretty much in this sort of central vein, and this they've actually the one that in their hands seems to behave the best is actually the Vienna library, which was being designed based around targeting conserved protein coding regions. And they've actually split this into two, so the average is pretty much the same as the other ones. But if you look at the ones that are the top 3 predictions of the guides that work, they seem to work better than the bottom 3 predictions. I think there's definitely differences between the libraries, and all of these, I should say, have been used quite successfully. Certainly this set in the middle here, but I think sort of it's certainly worth considering some of the Vienna ones as well as the ones from the Broad and the ones from Sanger as well. And I think the other thing to mention is that I talked about this idea of maybe using 5 guides per gene as a kind of standard starting point, and then people have tried to reduce that down in terms of viral, in terms of numbers of vectors that you need, and therefore the numbers of cells that you need. Now, it's worth highlighting that this is just an example from a screen that we've done, where this is actually the log-fold change of the 3 guides for this particular gene within that screen. And you can see that there's a good degree of variability between these different guides, even for the same gene. And this is perhaps a not very sort of well spoken about problem with a lot of these screens, is that in some cases these are sort of all in the same direction but just different in magnitude, but in some cases they actually have effects in opposite directions. And this is probably because one of the guides is not working, or one of the guides is having off-target effects, and that's causing loss in viability. So actually having this larger number of guides can be beneficial in terms of statistical power, but obviously it limits in terms of the cell types that you can use to do the screens. So it's a bit of a balancing act. I don't think there is a right and a wrong answer between what you should choose to use in terms of size of library. So I'm just going to highlight a couple where this has been sort of, one which I was involved a little bit in, which is this minimal library based on Cas9, where essentially we took all of the screens that were done with 5 guides per gene, actually across 3 different libraries. And we chose those guides that behaved most representatively. So not necessarily the ones that had the highest activity, because sometimes that can be due to off targets, but those that were most representative of the total for viability screens. And this means that you can then pick the best 2 guides per gene, and we found that 2 guides was the best. Going down to 1 seemed to cause problems, and going up beyond 2 didn't give us much benefit. And this is just comparing the top 2 that you see here with all 5 for that particular gene. And what you can see is that these are very well correlated with each other in terms of their viability. Actually, the 2 guides tend to have a slightly bigger effect size than the 5 guides on average, because these are being selected as being the best ones. And you get very sort of similar sort of precision recall of the essential genes. So this is incredibly powerful, I think, and allows you to do screens in more complex cells such as organoids, and that's ongoing work as well. But it does mean that the stats that you're doing post this on the analysis side of this, become less robust. So it sort of highlights the kind of importance of the follow-up screens and the follow-up experiments that go on. And a similar sort of process, this is actually this other form of sort of CRISPR-Cas, this Cas12a system, which actually naturally you can express these guides as an array with these direct repeats in between them. And that means that you can then express sort of 3 or 4 of these in one go without too much of a problem. And recently this, well, John Dench and some guys in Toronto actually have made a very minimal version of this kind of library where they've put 4 guides per gene. And actually for those genes which have paralogs, they've even put the kind of 2 guides for each paralog. So that it can show effects of those genes. And again, this behaves very well. Has the advantage that you also can deliver everything on one vector because this is quite a small Cas protein, so it isn't sort of quite as close to the packaging limit as the Cas9 would be. But again, in this case, we've only got 1 vector per gene. So whilst the size of the library is small, it also means that you are limited to just simply one knockout per gene. So again, the stats become a little bit more difficult, but it can still be used to identify essential genes, and it works pretty well in that case. There's also some constraints potentially with having this many double-strand breaks in a cell as well, which can be toxic to certain cell types. So what about if the cell numbers you can get are still too small to be able to use these genome-wide screens? And this can be in things like sort of organoids, differentiated cells, if you wanted to do anything with xenografts, which we'll touch on in a bit. Well, you can also make your own libraries, and I mean we do this quite regularly, and it's actually not such a difficult process to do. You order up these pools of oligos from various different suppliers, and then you can clone these in a single step as a pool into any vector that you like. And this gives you flexibility if you want to do something funky or you want to do something different with what you want to do. So it means you can use exactly the right vector for you, exactly the right sort of selection, the delivery method. You can use these slightly more complex systems such as dual guides to improve knockout efficiency. But I'd advise, if you are ever going to go about this, it's one of the most important things on these kind of custom library screens is actually to establish and include as many negative and positive controls that you know about as you can. And these can be in a variety of different types, anything from intergenic targeting controls, non-targeting biological controls, so that you actually can interpret the results that you get out the other end. So once you've decided on your library and your way of manipulating the genome, the next thing is actually to make sure that your system is actually working in terms of the activity of the Cas9 protein. And this is super critical again, particularly in these cases of sort of when you're looking for depletion, so sort of negative selection screens. Because if you have a certain proportion of unedited cells in there, then they will outgrow the ones that are edited, and therefore your sensitivity for detecting changes will be much, much lower. So you'll only pick out the absolutely top hits from the screen. And we found this to be this sort of reporter system to be super useful for this. So this is actually another lenti that you can put into your Cas9 cell line, for instance. And this contains a cherry marker to mark for those cells which have taken up the virus. And then a BFP guide that targets a BFP gene in the same construct. And this means that any cells that have taken up this lenti will be red. Any cells that are unedited will still be blue, and any cells that are edited will be not blue. So you can see this population here. This is the cherry axis. You can see this population is the transduced cells, and of those transduced cells, quite a large proportion of them in this case are actually BFP negative. So it says that the activity of the Cas9 is quite high. And typically we'll use a kind of a measurement of sort of somewhere above 80-85% cells being edited in order to actually be able to carry forward with that cell line. And it's also very useful because this is a highly quantitative and very sort of single cell readout. So it means that you can actually see the proportion of cells which actually would have an edit. You can also use endogenous genes as well, such as things like P2M, which is existing on involved in HLA presentation. So you can actually use this as a general sort of target for many different cell types. So moving on from the perturbations, now we're sort of into the cell models that you might want to do this in, and I think there's a huge sort of selection of this, sort of everything from what I've talked about, the sort of easiest probably, which is cancer cell lines, through sort of cancer organoids, primary cells such as T cells, iPS differentiated cells, which we do a lot of, even into things like primary organoids, xenografted cells that have been put back into mice, and even screens in vivo where you actually can deliver your viruses directly into things like the mouse. And these are very different in terms of how easy they are, and also in terms of the number of cells and the constraints which you might have. So things to sort of think about in terms of your particular experiment are cell numbers. So as I mentioned, there's a lot of cell losses, particularly, for instance, in the differentiation of iPS cells, there's particular cell losses. Bottlenecking. So in some cases, things like organoids will often be, when you passage them, will be passaged from fairly small numbers of cells. So it's more down to the number of cells that you're sort of passaging, rather than necessarily the total number of cells in the dish that will actually give you that coverage number that I talked about. The homogeneity of the final population. So if your cells are a mixture of lots of different cell types, that's going to muddy your readout. You obviously need to think about how you're going to deliver your reagent and whether you can make Cas9 cell lines and what would be your choice of vector that you need to put in. And also what your downstream assay is going to be. So you need this to have as big an effect size as you can. And also, in certain cases, like we do a lot of single cell readouts, and they can be very expensive. So it makes the choice of it makes the choice of the libraries very different to what you would do if you were looking at a growth phenotype. And then the third part of the screen is what you use as a readout, the simplest one is, as I've explained, these kind of life-death sort of assays, and this is definitely worth trying to, trying to couple sort of to any sort of readout. You can use reporters, for instance, with drug selections. You can also sort of just look at death in the presence or absence of a stressor. So this could be a drug, it could be a virus, it could be whatever you want. And it's also generally easier if you've got a positive selection. So one of the easiest screens to do is if you add a drug to a cell line and it kills all the cells, you can then screen for cells that survive and grow out of that, because that's super enriched for the knockouts that you're interested in. FACS is often a very good readout for many different phenotypes, and this can be, it involves a bit of development work up front, either for doing staining or reporter generation. You need to make sure that those effect sizes that you're hoping to see are as big as you can, and that it's reading out what you're expecting it to read out. And it does take a very long time if you're going to do a genome-wide screen to do the sorting. So sorting 50 million cells is actually quite a significant amount of time on a sorter. So you often will need to fix your cells, so your assay needs to be compatible with fixing, because otherwise it'll change during the time that you're doing the sorting. Again, I'll come back to it in a bit, but there's also down to the sort of bin size that you might actually use for your sorting. That's kind of a compromise between maintaining your coverage and getting the cell numbers that you need, but also seeing the effect size that you're hoping to see. We do quite a bit of single cell readouts, and this is very powerful because it gives you quite generalizable information on the transcriptome. This is just an example actually of a CRISPR screen, which we did with this sort of, I don't know, around 20 or so factors that we overexpressed. And you can see that these shift sort of quite sort of significantly in their transcriptome, depending on what gene you overexpress. And this gives you an enormous amount of information because you get sort of essentially 20 overexpression experiments all in one experiment. But it is quite expensive at the moment. And finally, I think it's worth just highlighting that this is possible, and it's an area that is certainly under active development worldwide, but also in our labs as well, which is this idea of pooled optical screening. So this is actually where you do imaging assays, which often you will have set up already for whatever phenotype you're interested in. And these imaging assays can then be coupled to detecting the guide RNA. This means that each cell you can detect the guide RNA in your imaging after your imaging readout. And that means that essentially you can apply this to any screen that you would like to in terms of anything that you have up and running that could have a microscopy-based readout. But this is quite challenging to set up at the moment. It's certainly not a sort of trivial exercise. But there are companies now sort of starting to offer this as a sort of machine, an instrument that might actually do this. So this is certainly something to keep an eye out for in the coming year or two. So the one that I'm going to focus on mainly is this fluorescent sort of flow sorting assays. I think one of the key things to kind of think about here, which is true of I think all different assays that you might use, is to make sure that your assay has the maximum effect size that you can imagine, and test this with controls of whatever you want to do. So this is really important to actually make sure that your screen would be a success. So this is an example where actually we fed neurons some protein, an aggregated protein called tau. And they take this up into the cells, when we knock out what we presume was the receptor for this protein, we can see a significant loss in the uptake. So this is kind of an ideal sort of assay because there's almost a separation between these two peaks. Whereas if you do this for many other assays, it's not so clear. So this is an ER stress assay where we're staining with an antibody for this protein called BIP. And you can see that this does shift in intensity when you add tunicamycin, but this is a bit of a sledgehammer. So when you do this with a gene knockout, the differences are much more subtle. And therefore, the effect size that you're seeing will determine the number of cells that you will need to do and the coverage in your screen, and therefore the difficulty in doing the screen. And the way I like to think about it is if you imagine you have a screen of 100 times coverage, what you're essentially having is 100 points within this light green graph and 100 points within this dark green graph. And would you believe, if you had 100 cells on your FACS graph, that there would be a difference between those two? And that's essentially what you're doing when you're thinking about coverage. So realistically, for most FACS screens, you'll need much more than 100 times coverage. The other thing that you can sort of consider is actually the size of your sorting bin. So typically what you will do when you're doing a screen is you'll sort the top X%, maybe the top 10%, the bottom 10% of your graph, and you can see that the difference sort of between the green, the number of cells within that green, the dark green component of this, and the light green component of this is enormous. So the effect size on that is quite large, but you're only sorting 10% of the cells that you're putting into the sorter, so your coverage goes down. So You could increase those bins to double the size, and then you would end up with double the number of cells. But equally, your difference between the dark green and the light green numbers is going to be less. So it's a balance between the sorting bin size and the effect size. And often what we've been doing is actually, because our sorter can do four-way sorts, we'll actually do 4 bins. We'll actually take the sort of the bottom 20, the mid 20, the top 20, and the slightly less top 20%. And that means that then you can you have the option of doing all the different pairwise comparisons and also looking across how things change across the different bins. So that's certainly something to consider when you're thinking about sort of doing these kind of screens. As I mentioned, you definitely need to fix, because otherwise the sorting time is just too long, and you know, for instance, in tau uptake, it would all be degraded and spat out of the cell by the time that you'd finished the sort. So this is important to make sure that your assay is compatible with fixation. And also, it's important to think about sort of replicates of screens. And as I mentioned before, there's this kind of difference between guides, but this is just an example to show the log-fold change of a set of genes across 2 repeats of the same assay. And you can see that a number of these are conserved and are consistent across the different screens, but there's also quite a few that are kind of stochastic outliers. And that's just simply because of the nature of the fact that you're only having 100 or sort of a few hundred times coverage of the library. It is at the end of the day a screen, and that's something which is worth bearing in mind. It's not actually giving you the answer. It's enriching for the genes that might be involved in that process. And then finally, there's a number of sort of computational methodologies that you need to do downstream. And I think probably the most generalizable and the most useful is this, and certainly the most widely used is this software called MAGEC. And essentially what this does is it measures the read counts in your, say, your upper and your lower bin. It does a log-fold change between those two. And then it ranks your guides according to that log-fold change after a bit of normalization. And what you end up with is this kind of ranking of all the guides that you have in your screen. And then if you were looking for a particular gene, you would say, well, okay, I'll highlight all of the guides that are targeting that gene. And that might be all of this sort of set of red guides. And these are generally enriched towards one end of that log-fold change spectrum. So you would say that this was an essential gene. And it uses a kind of enrichment towards sort of one end of that, a gene set enrichment analysis type stat to actually measure how skewed they are away from a kind of even distribution. You can also do that for pathways, so it doesn't just have to be genes, it can also be pathways. But this highlights that, you know, if you have more guides per gene, so really you need definitely more than 2, and sort of you know, something like 5 guides per gene this gives a much more robust result than it would do with 1 guide per gene, for instance. So you need to think about other ways of doing it with small numbers of vectors per gene. The other sort of class, if you like, that are sort of used are more these classification classes. So this is a bit of software called Bagel, which is a Bayesian sort of methodology, where essentially in this case you need your positive and your negative gene sets already identified. So it's mainly been used for essentiality screens because there is a set of core essential genes and a set of non-essential genes, and you can see that these exist in sort of certain positions on the log-fold change spectrum. And therefore you can actually sort of use that sort of those distributions to say, if I had a guide in this position, would it be most likely to be an essential gene or a non-essential gene? And you can use that to give you a p-value. So this again can be a slightly more sensitive way of doing this, but it does require this kind of positive control gene set in order to be able to be used. I think sort of, yeah, I mentioned this idea of sort of, feeding tau to these neurons, and I think this is a nice example of where we can actually of how you would actually go about a screen and also what you do afterwards as well. So this is actually an iPS-derived neurons, which then you feed, in this case, actually a mix of green labeled tau and red labeled transferrin as a control, and then you actually purify out the tau positive, transferrin positive versus tau negative versus transferrin negative neurons. So this identifies the kind of tau specific pathways of uptake. And you can do this with a couple of different substrates, and nicely it pulls out LRP1 as one of the top hits, which is a known receptor for the tau protein, and also interestingly this LRP2 gene as top of the hits. But what you actually end up with here really is a set of genes. If you actually take the top of this down to the significance threshold, you'll probably have 100 or 200 or 300 genes which are potentially involved in this process. Now, that's not an answer in terms of most biological questions. You can obviously look for pathways, and this is actually quite interesting because it pulls out particular pathways in the cell. But you need to have some way of actually getting from these hundreds of genes down to a few that you would be most interested in. And this is really worth thinking about when you're setting up these kind of screens, is what are you going to do downstream for your secondary sort of follow-up and even a tertiary follow-up. So in this case, what we did, this was actually a work with Lewis Evans, who's also at Sanger. We actually did basically a repeat of the FACS-based assay on a smaller set of the target genes, but doing the knockouts in individual wells, but still using the same assay to measure the output. So this validates, if you like, technically that those are real hits, and not just sort of spurious noise, because there is a false discovery rate associated with every primary screen. And then there was a kind of tertiary assay, which was more of a complex assay where we used imaging to do a time course of uptake. We used actually both knockout and drug inhibition experiments, and also tried different substrates that we were feeding the cells with. So that gives you a lot more biological insight into what's going on. And by this point, you've actually got down to a manageable number of genes that you're actually interested in to do those kind of assays on. So typically that's the kind of structure which we try to follow for these kind of screens. And it's definitely worth thinking about what these secondary and tertiary assays might be, because it determines how stringent you would be with the cutoff at the early stages, and not just cherry picking your top 2 or 3 hits. I'm going to spend the last few minutes just going through one example of a knockout screen that we've just completed, actually, literally sort of very, very recently, which is a knockout screen in iPS-derived microglial cells, which are kind of like the macrophages in the brain. This is the kind of process for this, which is quite a long-winded process, really, to go through this. It's kind of roughly sort of 3 months worth of work end to end, even if you know what you're doing. And as you'll see in a minute, there's actually been a number of points in this that we've had to work on to develop, and maybe highlight some points which are worth sort of thinking about when you're trying to set up this kind of thing. So we've actually done this and ended up doing this with a smallish knockout library, targeted knockout library, which we produced using our dual guide system to improve knockout efficiency, and an all-in-one vector to mean that we can deliver at any point in time. And we've also had to work on this way of infecting macrophages or microglial cells better than just adding simple lenti. So our differentiation for this, actually, you take iPS cells, you form them into little embryoid bodies using a 96-well round bottom plate, and then you seed those into flasks in a different media, and that generates these myeloid precursors that get spat out into the media. You collect those precursors and then you plate those down onto a substrate with a different cocktail of cytokines to differentiate into microglia. So the first thing that we thought about was actually where did we deliver the different components of the system. So we could deliver them at the early stage, or we could deliver them at the point of plating. They're probably the two main points to do this. And actually, this is a long-winded sort of story, but actually we've tried doing it at the early stage, and we find that actually there's a severe bottleneck at this embryoid body stage, in which you put 10,000 cells in an embryoid body, but actually only about 100 of them give rise to the remaining population of cells, which is quite a large number. So that means that it's very hard to introduce the library at this stage, because you'd need a kind of a room full of tissue culture flasks to actually be able to do this. Second is that we then tried to put in Cas9 at this stage and differentiate, but there's a problem with transgene silencing during the differentiation, so it tends to get turned off during this process. And the third is that when we tried to deliver virus at this point, the efficiency wasn't very high. So we actually ended up using a tweak that was suggested by a colleague that was working on HIV-2, which is actually co-transduction with a viral coat that provides this VPX protein that allows HIV-2 to infect macrophages. And that enabled us to be able to do the transduction at this point in time. So after a lot of optimization, we did that. And in order to mean that we didn't have to have the Cas9 in there, we actually used an all-in-one lentiviral vector. We also tweaked the sequence of the Cas9 to mean that we got better expression. And we used a dual guide sort of construct with two guides targeting the same gene within the same exon to give us a much improved knockout efficiency. So you can either get knockout from one guide or the second guide, or in the case of both of them working, you actually get a little deletion of the in between the two guides, which again maximizes the knockout efficiency. So now we got our Cas9 and guide components into the cells, and we got efficient knockout. We then had to sort of work on cell numbers, and this is, I think, sort of highlighting some of the sort of problems. We were initially trying to do this genome wide, but actually there's a lot of losses in the process. So first of all, we spent quite a long time improving the differentiation, and we increased the total number of cells that we can get out of the differentiation per week to be about the same as the number of cells that we put into it. This was originally more like 10% of what we put in. So the differentiation protocol we tweaked quite a bit. Then we used a published protocol to store these precursors over several weeks, because we can collect them over time, such that we can collect over about a month, and therefore we can maximize the number of precursors that we'd have. Then there's kind of losses in the transduction, as I mentioned, so only about 30% of the cells will be transduced. There's losses in the final differentiation of the cells. It's also worth highlighting that in this case we were using an assay where we fed the microglia a phagocytic cargo, which was actually dead neuronal cell line, and we actually had to add that at a 2-to-1 ratio. So we actually need double the number of neuronal cell line cells than we do microglial cells. So actually that's another sort of thing that we had to kind of bulk up. It's much easier to do, but it's certainly not an insignificant work. Then, of course, you're sorting 20% bins in our case. So that means that we only get 20% of those cells. And you get losses in both after sorting, some of the cells die, and also in the DNA extraction. And again, we improved that quite a lot by sort of little tweaks like increasing the incubation time in the proteinase K buffer to actually mean that we got a greater yield of DNA, particularly of fixed cells. So after all of this work, just to do sort of 1,000 guide library, which is what we were doing at 300 coverage for 3 repeats, we needed to start with 200 million precursors at this point in time. And that's completely copable with. But when we were trying to do this with the genome-wide library, we realized that actually this would involve something like 8 billion of these, and that's just infeasible to create. You need a room full of tissue culture incubators to do that. So this is, I think, just to highlight that it's not always this hard. This is actually a particularly difficult example of a screen that we've done. But it is certainly something that you need to think about in terms of the systems that you're using to do these screens in. And it isn't just a simple case of taking what works in a cancer cell line and applying it to your other systems. But after a lot of tissue culture, this was actually Sam with his enormous stack of tissue culture flasks, we did actually get a successful screen at the other end. What you can see here are the kind of genes which were showing increased phagocytosis or decreased phagocytosis when we knock them out. And actually, this is sort of new information, if you like. These are kind of completely novel biology that we're discovering here. We actually also identify known biology in here, so things like TREM2 is known biology. And in this case, we did actually do this kind of 4-bin sort, and that allows us to do things like this, where we can actually look at the kind of the counts across the different bins, and that gives you a lot more confidence that you're seeing a real effect, rather than just having 2 data points on this graph, which is actually how we identified the hits. And actually, we identify multiple components of the same signaling pathway. So actually, TREM2 is quite an interesting gene in terms of microglial biology and in terms of phagocytosis. And actually, in this case, it was quite unexpected, because actually the direction was opposite to what we would have predicted from the literature. But actually, all of the components in the pathway actually show the expected direction compared with TREM2. So these are both sort of positive regulators of the pathway, and this is a negative regulator of the pathway. So this is super consistent and also super consistent across different components of the same signaling pathway. And we were able to recapitulate that in arrayed assays. This is again with a FACS-based readout, where we can see that across multiple different cell lines. So this is an additional sort of thing that we added in our validation. We can see a loss in TREM2 protein when we put in knockouts, and also an increase in the phagocytic signal. And then our kind of, if you like, our tertiary screens were then we did knockout by different means. So this is actually using some published antisense oligos that downregulated TREM2 expression. And again, you can see this increase in phagocytosis signal. So this is all super consistent and means that we believe what's going on here. And we even went, actually, because we had a TREM2 knockout cell line, we actually looked at the effect of that knockout cell line. And again, you can see the same directionality of effect on phagocytosis. And interestingly, if we take the same cells and differentiate them into a different cell type, we actually see the opposite effect. So this is something we're now following up, and I think again just highlights the kind of power of these screens to actually uncover new biology. So okay, I think that was pretty much all I had to say, other than I think we're kind of heading in the CRISPR direction, and to thank everybody involved in this, particularly Sam, who did a lot of the microglial screening work, Sarah, who's been leading a lot of the sort of development work on the knockout sort of screen side, sort of to improve those screens, and of course a lot of other members of the lab who've been doing various other different components of this. So thank you very much, and yeah, happy to take any questions now, and also, as was mentioned earlier, I'm also happy to kind of for people to follow up with me afterwards, if that's kind of helpful, to avoid any pitfalls that we might have already gone through. So thank you very much.
Thank you, Andrew, for this wonderful lecture. It was really insightful. And now, before we proceed to the discussion, I would like to say just a few words how we organize this process. Everyone who is present here, you are welcome to ask your questions to Dr. Bassett. if you feel more comfortable asking a question in Ukrainian, it's absolutely okay. Just say what interests you. I will translate to Andrew. And you will get your answer. Also, for everyone who is watching the lecture online, please use the YouTube chat to type in your questions, and I will read it out to our lecturer. So please, now you have the chance to ask your questions. I will start with Alexander as the host.
Yeah, I'll use my position as a host to start. Andrew, thank you very much for your insightful talk. Actually, the question is a little bit diverged from the topic, but have you ever had experience with non-lentiviral based CRISPR screening systems? I'm specifically talking about virus-free systems like deriving of libraries using BXB recombinase, CRISPR recombinase, etc. What are pros of some systems and what of the limitations you actually see, whether they have them in comparison to the viral-based systems? Thank you.
Yeah, so it's something that we're actively working on actually, and we've been developing a recombinase landing pad system to which we're using for a variety of different purposes, but actually is one of the applications is for CRISPR screens as well. I think there's the advantages in it, I guess, are that you don't have to produce the lenti. And particularly if you're using these complex constructs like dual guides or prime editing sort of libraries or anything that has a kind of barcode separate from the guide, there's a big problem with template switching within the PCRs and particularly within the lentiviral prep, where actually there's recombination between different lentiviral genomes that can kind of separate the two ends of the virus. So if you've got guide A and guide B paired together, often there will be a recombination event that will result in guide A being paired to something else in the library. So it eliminates some of that issue. And also it means that, yeah, you actually can just produce plasmid libraries and transfect them into cells. So it's definitely possible. And I think the efficiencies are kind of the thing that is limiting at the moment. I think it's getting to a point where I think it's almost as good as it is with lenti. But there are certain caveats. So lenti is very nice because it can infect pretty much any cell. So if you're working with primary cells, for instance, it's going to be very hard for you to make a landing pad line to actually put your, well, impossible probably if you're working with something like a T cell. You can't actually create that line in order to be able to then put in your library. So it's therefore only applicable to certain cell types where you have a particular, where you have that ability to be able to produce those landing pad lines. So I think the answer is it can be super useful, but lenti can also be super useful, and both of them I think will still continue to be used. It's just dependent on the application.
So as a follow-up, if I understand correctly, that in case you're able to produce this cell lines that has a landing pad for the insertion, there is no other caveats in terms of the sensitivity for such screen, etc., to use such virus-free systems comparison to lentiviral-based systems?
I don't think so. I mean, I think there are also some advantages in terms of, because obviously lenti is a random integration. So not all of the lenti integrations will actually express, or some of them will be silenced over time. So I think there's no reason why it wouldn't be as good, if not better. It's just that it's a bit of a pain to make the lines in the first place.
And a last follow-up question, maybe because it's usually the problem for the laboratories that initiates at work, that lentiviral vectors actually require some confinement, like BSO2 laboratories that should be established to work on that. If laboratory will have some projects that need to implement CRISPR screens, and the only way for them is to dive into such virus-free systems, what are starting points of such system design would you recommend? Maybe you can recommend some of the architectures that works better better from your experience to try to implement that in OWN's project?
Yeah, I mean, I think probably get in touch is the answer. We're looking to write up something this year on our kind of landing pad structure. There are obviously some systems out there as well, and I think some of them certainly can work very well. So actually, even a single recombinase site is actually quite usable for screening, because you don't really mind about having the whole vector going in. And also, our system actually expresses a BXP1 recombinase on the landing pad, and that gets swapped out when you put in your vector. So it obviates the need to deliver the recombinase simultaneously with the library. But that makes it more complex to produce in the first place. So yeah, I think there's a lot of different structures that would work, but I think if there's particular interest, it requires a little bit more sort of detailed discussion, I think. So yeah, please do get in touch if you're interested, and we can share some stuff.
Yeah. Thank you for your question. Thank you for the great answer. We have some other questions in the audience, please.
Thank you. I have pretty theoretic question. Can we use, as I understand, we use in knocking out genes cultures of cancer cells? Is it right? Can we use other culture of cells, for instance, to examine inter-cells communications? We can knock out some synthesis of some cytokines or other molecules and try to understand do they involved in processes like apoptosis or cell communications or not? Can we do it in such way?
Yeah, so I think it depends on sort of what assay you'd like to use downstream. So if that signaling pathway would be involved in sort of regulating cell growth, then actually some of the screens that are already in existence might actually have the answers in them. So, for instance, we've done some work with interferon gamma signaling. So, actually, we can add interferon gamma, and that will kill off certain cell lines, and then you can look for knockouts that revert that process. But equally, if there's sort of certain signaling pathways that would be needed for cell viability, then they may well have already been screened as part of these kind of big cell line projects. But yeah, so certainly that's feasible. If you're looking for kind of non-cell autonomous effects, so things where you knock out in one cell and you're affecting a neighboring cell, that's a much more difficult thing to get into. So you can maybe knock out within a cell and then add another cell to it to actually stimulate it, if you like. That's kind of one approach, and people have done that for kind of T-cell sort of killing screens and that kind of thing. But it's actually quite difficult to kind of couple sort of knockout in one cell with effect in a neighboring cell. So that can be possible. It's something we're actually sort of thinking about, and lots of people are thinking about in terms of doing screens with imaging-based readouts, because then you can start to do that kind of thing. But yeah, definitely for cell autonomous phenotypes, you can work with it.
Thank you. Any more questions? Please here.
Yeah, so when you were mentioning the design of the vectors, you mentioned another enzyme, Cas12, and the only difference is that it's small in size, or are there any other differences in their enzymatic activities, and why did you choose Cas9 for your experiment and not Cas12 if it's smaller and maybe it's easier to maintain? So yeah.
Yeah, so I think part of the answer to that, it's a good question. Part of the answer to the question is actually historic. So Cas9 was one of the, or a strep pyogenes Cas9 should have had, is actually one of the earliest enzymes to be found, and actually seems to work probably one of the best still, actually, no matter what anything has happened in between. You're right, Cas12 is smaller. I think the other advantage is that it has these kind of, you can put in these repetitive sort of sequences that can allow you to express multiple guides without any kind of tweaking. I guess the downsides are that we don't understand the rules behind which guides work well as much, because that hasn't been as well studied. And actually, in general, for a single guide cut, it seems to be slightly less efficient than a Cas9 would be. So that's one of the reasons why they've gone to 4 guides per gene, is so that they recover that loss in efficiency that you see with Cas12, even with a lot of tweaks. Originally, it didn't work very well at all, but there's been a lot of work done by Ben Kleinsteiber and John Dench, who've kind of improved those a lot. But even still, after those improvements, I think with a single guide direct comparison between the two, the Cas9 still works better. And the third thing is just logistical. You know, we have a lot of Cas9 cell lines. You know, there's 800 Cas9 cell lines. So if we don't have to reinvent the wheel, then we can reuse those for other purposes.
Yeah, thank you very much for your answer.
Thank you. Any more questions? I might not see the whole audience. Yes, there is. Yep.
Firstly, thank you for the lecture, and secondly, what programs or program have you been using to analyze and build the graphics and diagrams of the data you used, and were they free?
Yeah, so I think it's a massive mixture. So a lot of the I think the analysis of the CRISPR screens is mainly MAGEC that we use, which is definitely open source free. Most of the plots are done in R in a variety of different bits of variety of different packages. Again, all sort of open source and free. There might be the odd sort of figures that have been put together there that actually have just been assembled from component parts of various other ones in other software, you know, Illustrator and things. But yeah, I think most of the plots are done in R, and often are part of the packages that come with it. So actually, MAGEC is quite good at sort of having a lot of different plots as part of that package. So yeah, a lot of those sort of, you know, for instance, the guide specific plots and the sort of the waterfall type sort of diagram, they're all things from the MAGEC package.
Thank you so much.
Thank you.
Yeah.
We have a question here.
I'm sorry, I have very ridiculous question. How many oncogene today we can to edit? And what about clinical application of this technology?
Yeah, it's a good question. I mean, I think I think clinically it's tricky because you're kind of still reliant on drugs in the main. So I think CRISPR application in vivo is still in its infancy, and actually delivery into these kind of tissues is quite difficult. So it's probably more to do with kind of identifying targets that are then drugged in a more classical manner. I think there's a lot of candidates from these screens, but there's an awful lot of work that goes into taking those candidates into actually sort of following up and double checking. So from the DepMap screens, there's probably around about 5,000 genes that are essential in at least one cell line. And of those, there's probably a few hundred that would be good candidates. But it's then kind of, what do you do with those downstream? And actually to follow up on even one of those, like Werner's, has been a major effort. You know, that's even just the kind of validation that really is true is quite tricky. So that's prior to actually developing the drugs against it. So I think there's still a lot of need for people to fill in that kind of intermediate space as well.
Thank you. Any more questions? Yes, please, here.
I want to ask you about the construct where you used two guide RNAs divided with tRNA. So why do you use tRNA in this case, and can anything else be used in similar construct?
Yeah, so I mean, there's a lot of different versions of these kind of things that people have played around with. We did actually quite a lot of work to settle on the tRNA structure that we have, which is on bioRxiv. All the sequences are there now if you're at all interested. So one reason is that the tRNA machinery is present in virtually every human cell. So and it's nuclear and it's very abundant. So that means that processing happens very efficiently and very effectively in any cell. It doesn't add any extra extraneous sequence. So that's important because otherwise you'd end up with interfering with guide activity. It's also quite fortuitous, actually, that the tRNA doesn't just act as a cleavage signal. It also has its own promoter activity. And the sum of the promoter activity from the tRNA and the cleaved U6 actually turns out to be very well balanced between position 1 and position 2 within the construct. So actually the second guide is boosted up, if you like, by that tRNA promoter activity. So it means that we get a very, very good balance between the two. There are a number of other systems. I mean, having said that, there's a number of systems out there that people have used, ribozymes and there's a little structural unit which is a binding site for CSY4, which is a nuclease that cuts. That requires then expression of the CSY4 nuclease, though, and none of the other systems that are kind of based on ribozymes or that kind of thing have actually been very effective in terms of how efficient they are in terms of cleavage. So I think it is the best way to go in terms of Cas9 pairs. And I think now with all the tweaks that we've done to the sequence of that, I think it's now very well balanced between the two, which is often a bit of an issue with even two promoter systems which people have used. And those tend to be slightly unevenly balanced between the two positions, and also have issues with some of this template switching that I was talking about, because the two guides are a long way apart, and also because of the fact that you have to do a two-step cloning process, whereas ours is short enough to fit onto one construct. So I think it's a really nice system actually to use, but I would say that.
Does it really matter which kind of tRNA it is, or just a random one?
It does. We've been through, it's not actually, we didn't actually bother putting it in the preprint, but we went through a few different tRNAs, and they are different in their activity. It happens to be that the one that we first tried, the glycine tRNA from humans, seems to have the best activity, but there are a couple of others that can be used, but they're slightly less efficient, and not all tRNAs are the same. Some of them are awful.
Thank you very much.
Thank you. Any more questions? Maybe I will use my chance to ask a question too. You talked about the knockout libraries available, and we are looking into performing activator screens. So my question is whether maybe you have some tips on which software or other tools to use to actually design single guide RNAs or guide RNA for CRISPR for Cas12a enzyme libraries to use in activator screens, and maybe you have some tips on which sites in promoters. Maybe if there are any tips in that regard which work better or worse, maybe in regard to the TSS site, just where to look into.
Yeah, I mean, yeah. So I guess from a Cas9 point of view for activators, there's two sides. So one is the library, and I think the most recent Weissman CRISPR-A libraries are very good, actually. They're much better than some of the previous generations. There's a huge variability between different guide positions in terms of their efficacy at activating. And one of the tweaks in that recent one is, first of all, to use a machine learning approach to decide on the guides, but also to pair them into pairs. And both of those seem to improve the efficacy of that quite a bit. Second is actually the effector domain that you attach onto the Cas9. And again, there's been a number of iterations of that over the years. The one that we've had most experience with is called VPR, which is 3 different transcriptional activators directly fused to Cas9. That works quite well in our hands. The SAM system is an even more complex one where there's actually a stem loop, an MS2 stem loop on the guide that then recruits different factors. And that, again, will probably work better. It'll give a higher level of activation, but it's difficult to get the balance between the effector MS2 coat protein fusions and the Cas9 fusions. So it's a little bit more finicky to set up in the first place, but probably better when it actually works. So in terms of what's existing and available, I definitely look into those. There's also some tweaks to the VPR type architecture that have been published recently that again we haven't tested, but I think would be worth a look at, because I don't think it's not without a little bit of toxicity in that process as well. In terms of position relative to the TSS, I think most people agree that sort of for CRISPR-I, targeting around the TSS, which I would define not by the end of an RNA-seq transcript, but by the cage data from Riken, for instance, that seems to be the best, because you get spreading of the repression within sort of maybe 300 or 400 bases, and also some interference with the basal transcription machinery binding if you target around the TSS. With CRISPR-A, it seems to be better to go slightly further upstream, because you're not then interfering with the basal transcription machinery binding, but you are recruiting the activation sort of components upstream. So again, there's kind of probably a little window slightly more upstream where it's better. And in terms of Cas12, I think there's probably less known, and I think it can have its advantages in terms of the sequences in and around there might be more amenable to Cas12 targeting, but it is difficult with any of these dead Cas9 versions in terms of off targeting, because you don't require quite as long length to bind as you do to cut. So there seems to be a binding with a relatively small section of the guide, but only when you get base pairing across the whole length of it do then you get cutting. So there's a little bit more, they're a little bit more promiscuous in terms of their off targeting. So I think designing a Cas12a CRISPR-A library would be quite challenging. I think you need to go for lots of guides, is my advice, probably in pairs. We've done some work with putting in up to 5 guides, and we see a kind of increase in CRISPR-A activity up until about 3, and then it kind of you don't get much benefit. But that's with Cas9. So having multiple guides within the same cell is definitely beneficial for that. Just increases the number of copies of the activation domains, I think.
Thank you. And a follow-up question on what you have just said. In terms of activation efficiency, from your experience, what is the average fold change at which we are looking at?
It's very hard to say. It can be anything from nothing to sort of 1,000 or 10,000 times. I think so from our experience, we did a bit of work with a standardized reporter that was actually integrated in lots of different chromatin sort of states. And what we found there was actually that the basal level determined the activation ability. So basically genes, or it's actually the same reporter gene with the same guides targeting that reporter. If it was high expressed, it tended to be more difficult to activate more than if it was low expressed. And there is some chromatin dependence of that. We have a paper a couple of years ago about that as well. But most of the difference in the activation efficacy came from the basal level of expression of the gene. So broadly, the higher a gene is expressed in your cell type, the harder it is to activate, or probably because it's already got a load of activation factors sitting there. But it seemed to be quite robust to chromatin structures. So I think there's probably some, there's a lot of feedback mechanisms as well, sort of downstream. Some genes are much more tightly controlled at the RNA or protein level than just simply boosting up transcription will do. So I think there's a lot of layers of regulation there, but yeah, that's definitely a general feature.
Thank you. Maybe anyone else has a question? Okay, I see over there.
Thank you for your lecture. It was great. I'd like to ask, for example, if there's an experiment where different CRISPR-A options are evaluated and compared, would it be better to use a single guide RNA for this experiment or a bunch of them, CRISPR-A, to evaluate different CRISPR activators? Or it's better to stick to one to avoid less variability in experiment?
Yeah, I think if you want to, so I think different activators have different functions with different genes. So certain
Sorry to
activators
interrupt.
work
I'm
better
talking
at
about
certain
one
genes.
gene and different activators for evaluation on one gene. Just
Yeah,
these
I would be
activators, yeah.
reluctant to go for a single guide. I think it depends on what you want to try and get out of it. If you're just looking at sort of activator efficacy with that particular guide, you can obviously see changes if you use a single guide. But I would worry a little bit that that's not going to become generalizable when you try to apply that across other genes. I'm just trying to think if there's any evidence that we have of that. Yeah, I think actually we did some work with P300 as a fusion to Cas9, and that certainly didn't behave. It was guide dependent how well it worked. So if you compared it with sort of some of the others like VPR, that was more consistent, I think, across multiple different guides, whereas P300 was very much gene specific. So when you targeted with a different guide or a different sort of targeted a different gene, it was variable, whereas the VPR was much more consistent. So yeah, I think you probably need to go for multiple guides across multiple activators to really get a feel for what's going on.
Thank you very much.
Thank you. And we have a question from the online audience. Thank you. You mentioned in vivo models in mice. Have you done some experiments with in vivo models?
We haven't personally, no. I know that sort of there's a number of groups worldwide doing this, and I actually went to a meeting the last week where there's some very exciting data using AAV to deliver into neurons in the brain of mice directly to do pooled screens with then a single cell readout at the other end. So I think it's super powerful, and actually you get quite a lot of cells in a in vivo model. But there is there's a whole host of kind of sort of hurdles, I think, to overcome. So I definitely encourage you to talk to people who've done it before, because I think it's not a trivial thing to set up.
Thank you. Any more questions? Over here, please. Yeah.
Yeah, first of all, Dr. Bassett, I want to give a huge shout out to the team performing all the cell culture work. It's really impressive. I can imagine it. And I have a question. When we are talking about knockout screens, especially in cancer cells or cell lines, typically those cells have like genomic rearrangement events are typical for those kind of cells. So when we're talking about like small indels, or even if it's like between two guide RNAs, you have like small deletion. So could it be confounding when these cells undergo some sort of rearrangement? And so when you have this guide screened in the cell, but you don't see the phenotype just because the edit was re-edited, let's say it.
Yes. Yeah, no, it's a very good point, and one I didn't actually touch on, but even within the sort of DepMap screens, there was a big issue with regions of the genome where there was kind of, maybe tetraploid or hexaploid sort of copy number amplifications. They found there was actually just targeting those regions with a guide caused the cells to die. Probably because you're getting multiple cuts within the same cell over the same genomic region. And actually the analytical processes downstream had to be tweaked to adjust for that. Because what you could basically use is you could actually use it to map these copy number variants, because you could see all of the guides within that region were actually causing a depletion. And you had to separate that from the effect of the individual genes within that region on the phenotype. So that was definitely a confounding effect and could impact on it. The other thing is also, if you've got more copies, then you've got more copies to knock out, because obviously to get most of the phenotypes you're looking for, you need a homozygous knockout. So it becomes harder as you have more copies to delete. So I think there's definitely some issues there. And then the third thing is also targeting towards the end of chromosomes. Sometimes you end up with the end of the chromosome falling off if it's not sort of particularly essential. There's no sort of major genes in there, and that again can confound some of the results you see. So yeah, it's very good to be open-eyed when you're going into these things and realize what the context is that you're doing the screen in. So yes, is the short answer, but there's a long one there as well.
Thank you. And the one rather technical question, sorry if I missed it. When you mentioned the optical screens for the phenotype, you mentioned that you can identify the gRNA that caused that phenotype. How it's technically made after the optical screen?
Yeah, so it's a process called in situ sequencing. It's essentially you have padlock probes that bind to the RNA or cDNA, doesn't matter. And those padlock probes, essentially, you can either ligate them when they're bound, and then that allows rolling circle amplification with 529 polymerase, and then you detect those with a barcode and a series of cyclic hybridization and stripping reactions. So you can imagine if you've only got 4 fluorescent colors, you can only detect 4 different species. So this is a way of basically using a series of cycles to then be able to detect multiple hundreds or even thousands of different transcripts or different guides. And then there's kind of a second strategy. So we're using actually that kind of strategy with barcodes in the constructs. But there's a second one which is exemplified by Paul Blaney's work, where they actually have a gap at the bottom of the padlock probe that they then fill in, and that gap actually turns out to be the guide RNA sequence itself. And therefore you can fill that in with a polymerase, then amplify that up, and then actually do sequencing across that region to identify the so you do MySeq basically in situ in cells. And that's actually what the methodology that the element sort of sequencer that I was sort of mentioning as the kind of commercialized version of this is working towards as well. So yeah, it's complicated and probably I haven't explained it very well on the kind of simple level, but broadly you essentially are using the RNA expression to detect the guide RNA in each cell.
Thank you very much.
Thank you. There was a question here.
Thank you so much. That was a really great talk. So I was also curious about the long-term steps in your experiments. So the in vivo part, and just out of the curiosity, in case you need to deliver your editing system to the brain, and you want it to go through the blood-brain barrier, what would you use for that? Just hypothetically.
Yeah, I think it's quite a tough sort of thing to do. I think most people are actually injecting it directly into the brain rather than trying to go across the blood-brain barrier. I think there's a whole field working on that kind of thing, and I know very little about it, I must admit. But there are various different receptors that people have used, so you can bind to those receptors, and it helps to kind of transfer across the blood-brain barrier. But I must admit, I don't know much of the details. So,
thanks.
Thank you. One more question here.
Yes, thank you very much. Dr. Sprice, you already mentioned some of the control constructs you usually use with the Cas9 knockout systems to assess activity of the systems in different cell types. Could you recommend some of such type of control constructs to assess activator systems in different cell types that will be predictive and representative for the actual performance of activator systems? Thank you.
Yeah, I'm not sure the reporter constructs have been terribly effective, actually. I think probably the best thing is to find a gene that you can measure on the cell surface or sort in some way or other, and then use that as a measure of how effectively it's working. So, for instance, I talked about the B2M knockout as being a way of detecting knockout efficiency. I think you could easily sort of overexpress a receptor that is not normally expressed on that cell type, a CD something or other that you have good antibodies to, and then that will allow you to then be able to detect the activation ability. So that would be my suggestion, but I think reporter systems haven't been terribly successful. So.
Thank you. One more question over there.
Why lentiviruses are so popular in regards to CRISPR, and would it be safer for humans to use vectors which do not integrate in human genome? And have you worked with those other virus vectors?
Yeah, so I think for screening, I think the integration in the genome is important because we need a way of marking the cells that have a particular knockout. So that's kind of an essential component. I know that people are working with AAV, for instance, and have done that, have been able to get that to work, because there's kind of an episomal expression of the guides. So that might be a possibility. I think, as you say, if anything that you were doing in humans, you probably wouldn't want to do with lenti, because of the integration but that's kind of, I guess, a slightly separate problem from the screening problem. So yeah, that's kind of the major reason for using lenti is to mean that it does integrate, and therefore you have this way of following the mutants over time.
Thank you. I might have a little follow-up question to this. In terms of stable expression of Cas9 nuclease in your cells, Is it a rule of thumb to use an inducible promoter, or it could be a constitutive promoter to express the nuclease in the cells?
Yeah, so I think most has been done with a constitutive promoter, actually. The inducible promoters can work, but we've had real problems with transgene silencing in iPS differentiation, for instance, and that's meant that that's even worse with an inducible promoter. So we tend to avoid it where we can, and I think it's best to keep it as simple as you can. Actually, there isn't much effect of overexpression of Cas9. You can generate an overexpressing Cas9 mouse constitutively, and it has very slightly non-Mendelian inheritance, but it's not. There's no overt phenotypes. It seems to be perfectly fine. So I don't think there's a very big effect of Cas9 by itself. The other thing that you have problems with inducible systems is actually getting the balance between the on and the off rate. And typically in our experience, we either have a background expression or we have a reduced on expression, and neither of which is very good, because generally you want to keep it off if you want an inducible system until you want to induce it. Or you want, but also you want to have maximal activity, and therefore there's kind of there's a balance to be hit there. The only exception I think is kind of been in base editors where there is some toxicity from overexpressing constitutively, and therefore we've had better success with inducible systems in cancer cells, but not so much in iPS cells. That seems to still be a problem.
Thank you. Thank you. Maybe one last question over there, please.
Is this technology suitable for determining genes that works when the cell have 2 copies of healthy gene?
Yes. So I think yeah. So most of the technologies I presented, knockout, CRISPR-I, CRISPR-A, are all perfectly feasible in diploid cells, even in higher ploidy cells. So some of the cancer cell lines are kind of triploid, tetraploid in the main. So yes, is the short answer. You can definitely use it. And it was something that was slightly surprising, I think, when they first tried this, that it actually worked quite well in many cells. But I think it's because you actually, certainly with the knockouts, you actually accumulate over time the mutations. So they don't just sort of, any unedited allele will edit again. And so if you leave it in for a week, then almost every allele within the cell will become edited.
Thank you. Thank you for the great lecture. It really provided a lot of insights, and I think it raised many questions in our minds, and there definitely will be people coming back to you with their specific questions. So if you don't mind, we will provide them with the information necessary to contact you.
Oh, that's great. That's wonderful. And yeah, it's nice to have such an engaged audience as well. It's great to sort of great to hear all your questions, and they're sort of yeah. Hopefully, I can avoid some of the pitfalls that you may have come across, and if I can do that, then that's wonderful, because it saves a lot of people a lot of time. So, thank you.
Thank you for your time, and we hope you have a great evening. And now I give the floor back to our hosts.
Thank you, Dr. Bassett, Dr. Szenderowska, and all participants who joined this lecture and such an engaging discussion. It was really very interesting. I really thank you, every one of you. And of course, CRISPR libraries are powerful technology, and we hope that its implementation of this technology will help you to tackle your toughest questions and working on. And many thanks that Dr. Bassett and all of you and all our partners helped us to start BioGENext series. But it is just the beginning of the whole series.
Yeah, and our second lecture actually will cover even equally important methodological slides, and we'll give you practical intro into the protein structural biology. Actually, it's a Nobel-winning topic last year, and actually recently, due to tremendous advance in computational and experimental structural biology in recent years, researchers actually gained access to a vast amount of structural data, modeling data, also based not only on conventional tools but on AI-based tools, and actually the ability to really understand the data, understand its quality, evaluate its quality, understand some of the key results, and derive from the data answers to biological questions is a paramount important. It's a skills that have paramount importance for modern life scientists.
In a month, we will introduce to you our next speaker, Mykola Protopopov, the head of computational chemistry at ChemSpace, and his lecture, his talk will address the practical aspects of protein structure, modeling, structure interpretation, and structure-driven drug design. The lecture will be hosted at 212 Hall at the of Institute of Biology and Medicine campus of Kyiv National Taras Shevchenko University. The cold in winter, but heartwarming auditorium for many university students and alumni. The registration for this event is now open, so please use the QR code provided here and be among the first to reserve the spot. And follow the BioGENext initiative on social networks and BioGENext event series on Eventbrite portal to receive all updates about the upcoming series events.
Yeah, and what is actually? Yeah. And what is actually I want to add, our aim as an initiative is actually to bring as much as it's possible expertise here back to this audience, and we will encourage and try to bring as much speakers as it is possible, actually practically on-site, offline, and Mykola will be one of such speakers, so he will be on-site. You will be able to just grab him after lecture and ask every dirty question you want from him in field of structural biology, actually.
Don't tell him if you know him.
Yeah. So actually ask every question you want directly to the speaker in order to obtain your answers and make a first steps in order to use all of that amount of data in structural biology you already have or can have access to. So with that, thank all of you for joining us today, and hope you will have a great time later to meet your old friends or meet your new partners later in the networking session we will have, and which will follow the lecture. And have a great evening, and see you soon at next BioGENext series events. So thank you all for being here. Yeah. For the networking session, we have a time to approximately 8:40, but we will see how the hosts will get out also there. So you will have time at least before that, and we will like very much to see all of you during the networking session. Thank you very much for being here.